Uh, Good morning, everybody. I'm not a ghost. Yeah, I have not appeared at any such meeting, ISMB, PSB, Recomb, ECCB, since 2013. So the most people thought I disappeared. But this is a real organism, and I'm not a ghost. Today, well, this is the, okay, shit. Today's topic is about cause. Some of the written in 3 letters. Okay. Well, AI is climbing up the ladder. That ladder is going towards high intelligence. And first, prediction models and explainable AI, and now causal AI. Cause is a very important topic in AI, especially in biomedical field, especially, healthcare and medicine. Well, the world is going up to this ladder, but somebody is falling down. Yeah. Well, I made this picture by using Gemini, and my face photo is added there. Okay. Causes of cancer is mutations on genomes, as you know, and also aberrations on epigenomes and so on. Yeah. So on genome, some T is changed to C, and some region is deleted, or some region have some insertions and so on. And this causes the system aberrations, but most of the cells with this kind of aberrations are killed by T cells or die with a system of apoptosis. But the cells escaped from those barrier is cancer cells. And cancer was recognized as cause of cancer was identified around 2070s. This was discovered not for human, but this nominal paper shows the cause of cancer is genome mutations. So the understanding cancer was or is search for mutations on genomes from cancer cells. But currently, only looking at cancer cells is not enough to understand cancer. We need to understand the non-cancer tissues together with cancer cells around. So the cancer cells and the non-cancer cells are coordinating to support cancer to survive. This is a current understanding. Well, the comprehensive search for mutations is very important. In books, mutations are like that. Some insect bites and make a hole and so on. Yeah. Our genomes, especially cancer genomes, have these kind of aberrations. Well, I want to talk 3 stories I faced with. The first challenge, my research history is very similar to Dr. Eric Green. 35 years from the beginning of Human Genome Project around 1990. After completing the human genome, then I got into cancer genomics. And this is the first challenge in collaboration with a top cancer biologist. That happens. We started 2010. This is a story for these 2 years. Well, some of you remember that we had a serious disaster that is called 3.11 in 2011. Yeah, it's a great East Japan earthquake, tsunami, and almost 20,000 people died. And also, in addition, Fukushima nuclear weapon plant disaster is still a big problem in Japan. But in 2011, we faced with a serious problem about research. The limited use of electricity. So some area, the electricity was planned to stop. For example, maybe in this meeting, Dr. Asai, could you raise your hand? Oh, well. He is at the University of Tokyo, but in a different campus from the Hongo or my campus. And his campus lost electricity for some period. It's a planned. So that period, no computer, no sequencer. And one run of sequencer took 2 weeks at that time. Currently, 1 day is enough. That means we have continuous vibration that prevent us from sequencing. So it stops sequencers and also stops supercomputers. Unfortunately, the campus where Human Genome Center is really in the center, near to the government. So we needed to reduce the use of electricity, but we could run with a limited amount. And during that disaster, we challenged to solve the problem of MDS. MDS is myelodysplastic syndrome. And bone marrow fails to produce enough healthy blood cells. People with the age more than 60 has a high risk of facing with we call MDS. And the common symptom is, anemia and so on. Well, people think it's not a serious disease, but 30% of MDS patients move to AML, acute myeloid leukemia. That is near to death. But the cause of MDS was not known at that time. And at that time, in collaboration with Professor Seishi Ogawa at Kyoto University, at that time he was at the University of Tokyo. He is a hematologist and cancer biologist. And the samples we used is 29 MDS samples for comprehensive mutation search. Sequencer was Illumina HiSec 2000, SNP array-based copy number analysis, RNA sequences, RNA microarray data, and 582 MDS patients, which were used to validation for mutations we found. And the computer we used was, we call Shirokane at Human Genome Center. And it has just 6,000 CPU cores, and the storage was 1 petabyte raster file system. At that time, this was the limit of our capacity to build. And on that computer, we developed a system to analyze sequencing. We call Genomon Go, get mutations. And it's a suite for bioinformatics tools for analyzing cancer genomes and RNA sequence data. This was developed by Yuichi Shiraishi and Keisuke Kataoka and Seishi Ogawa's group. Yuichi Shiraishi was at that time my postdoc there. Well, 15 seconds promotion. This is assembly process. RNA sometimes help. That's it. Okay. And we sequenced just 29 MDS patient samples, carefully selected by Dr. Ogawa. And we sequenced not whole genome because of the money. We couldn't have so much money to sequence whole genomes. And from 29 samples, we got 268 somatic mutations. And we listed those mutations with respect to frequency. Then 4 genes appear, not in the top, but in higher than the middle. And those genes are RNA splicing factors. And at that time, hematologists and oncologists didn't recognize why these RNA splicing factors appear in MDS patient samples. Then, well, these 4 mutations related to RNA splicing was validated by using 582 patient samples, and also the mutation-induced HeLa cell, which result in abnormal RNA splicing. This was used by mice, and also validation of hematopoietic function by bone marrow transplantation to mice. Well, then we reached the conclusion, well, these 4 RNA splicing factors aberrations are the cause of MDS. It took 1 year. We started in July 2010, and we could publish the results September of 2011. Well, it was a very, very severe period because of the lack of electricity, and also, well, Japan is completely sunk. And this was introduced by Eric Landa, at that time he was a director of Broad Institute, in the meeting in 2011, November, he mentioned that this is the first story that shows abnormalities in RNA splicing are involved in the development of cancer. Okay, next, evading cancer, evading immunity. And this challenge was done in 2006, and also before that, the whole genome sequencing and omics analysis for finding the cause of evade immunity. Well, at that time, we could use K-computer at RIKEN. This achieved the #1 in 2 periods. And simultaneously, we run the program at our Human Genome Center's Shirokane. And then first comes, we appreciate. And the disease is, maybe most of you don't know, it's an adult T-cell leukemia lymphoma. This is a very local disease in the western part of Japan. Cause is known. It is caused by infection of adult T-cell leukemia virus, HTLV-1. At the age of baby is infected from the mother by and extremely aggressive. When appeared, then maybe around 6 months, the patient will die. We collected from Japan in total 426 samples, and 48 samples was whole genome sequenced. Short read, at that time, 100 length. And whole exome analysis, and RNA sequencing, methylation analysis, and validation were done with 343 samples. And the genome pipeline discovered from the 48 whole genome sequencing data, almost 3,000 structural variations. 1,000 is very many, but 3,000 is so many. But young scientists looked into these aberrations and created a map of its agony, map of major driver alterations. It's a battlefield of T cell and HTLV-1. And interestingly, immune checkpoint pathway have aberrations. That aberrations are not single nucleotide aberration, but structural variations. Well, maybe I will explain a little bit about how cancer evades immunity. Well, T cell is something like a police, catching cancer cells and kill it. And the cancer cell shows antigen, and the T cell police find that and kill it. However, cancer cell, not only cancer cell, but normal cell too, has a system to produce a protein called PD-L1. It's something like a bribe money to put into the policeman's pocket. And that policeman's pocket is PD-1. This was discovered by Professor Honjo at the Kyoto University. He is a Nobel Prize laureate because of the finding of PD-1. Well, meaning that cancer cell has the ability to produce bribe money, and those bribe money are put into Professor Honjo's PD-1 bribe money receiving pocket. Then police doesn't kill it. Stop. The drug inhibiting, so the bribe money's pocket is seized, then bribe money doesn't get into. This is a mechanism of Nivolumab. But this mechanism was known. However, no genetic aberrations were known about that. We sequenced 48. Then, interestingly, the genetic aberrations of PD-L1, bribe money production system, in ATL samples with SVN, structural variations involving PD-L1 3' UTR, non-coding regions. Non-coding region structural variation, which are not transcribed to RNA. Well, it's a transcribed to RNA, but not to protein, has high mRNA expression of PD-L1. That means PD-L1 bribe money are produced a lot. And that mechanism was validated by experiment, and the 3' UTR structural variations produce RNA with which the mRNA, PD-L1 mRNA, are not degraded. That means a lot of PD-L1 bribe money were produced. And this was validated experimentally. But this is about ATL, local disease. Then we analyzed the more than 10,000 cancers ranging over 33 cancer types. Very rare, but in different cancer, this kind of aberrations were found. So this was a story of genetic cause, how cancer evades immunity. This research is heavily depending on computing. The last is COVID-19, 2020 COVID-19 pandemic. And in Japan, two questions. The comparatively small number of severe patients in Japan. Why? And genetically, why? And which genetic factors in severe patients in Japan? Young patients also have a very severe, critical severity. And another question is, can network biology unravel causes of COVID-19 severity? And Japan, in order to cope with that, we formed the Japan COVID-19 task force by volunteer people. Takanori Kanai, he is and Koichi Fukunaga, he is at Keio University, and Seisho Ogawa, my long-term collaborator, and I discussed what we can do by Zoom meeting almost every 3 nights. And we decided to form a task force by writing emails to possible people, and 120 hospitals agreed to collaborate. And finally, we collected more than 6,500 patient samples. It's a peripheral blood that collected. And from peripheral blood, DNA, RNA, serum, and plasma are separated and stored, and we analyzed. Well, at that time, supercomputer Shirokane is evolved. Shirokane 5, Shirokane 6, Shirokane 7. And most people don't know what is Shirokane. Shirokane is a name of the place where Human Genome Center is. Its location is there. Yeah. And at the stage, we could collect around 3,000 patient samples. And first, we sequenced all 6,000, 5,000 patient sequence. Whole genome, whole RNA sequences are done. But at the stage of almost 3,000 patient samples are collected. And then GBAS analysis was done. And at that time, 3,000, but finally, this number, the non-severe patients, around 3,000. The GBAS analysis was done, simple work, and doesn't require so much computation, but so much human labor to find the specific allele. And we could find the variant on chromosome 5 close to the dedicator of cytokine kinase 2 gene, DOC2. This was discovered by a professor at Kyushu University, is associated with severe COVID-19 in patients less than 65 years age. And RNA sequencing analysis of 473 bulk peripheral blood samples identified decreased expression of DOC2 associated with a risk allele in these younger patients. And the DOC2 expression was suppressed in patients with severe causes of COVID-19. Experimentally, hamster was used to validate this one. So, and at the time, we could use Fugaku supercomputer. Fugaku was used for disease network biology. For example, Bayesian network estimation requires a huge amount of computing time, but Fugaku provides easy solution to that. And Fugaku achieved #1 for period and for top 500, and graph 500. It has a very long history of #1. And the severity hubs associated with COVID-19 were unraveled from inflammatory-related RNA protein networks. While the inflammatory response are central to COVID-19 severity, the understanding of molecular mechanism, particularly the interaction and hub structure with RNA protein network, have not been fully elucidated. And from the COVID-19 task force, Japan COVID-19 task force patient samples, the RNA data from almost 1,000 genotyped samples, and the protein expression data. This was obtained by O-link method. And the protein and RNA network are computed by using Fugak supercomputer and also Human Genome Center's supercomputer. Then, this is very complicated. The atypical chemokine receptor 2, ARC2, network analysis showed ARC2 acts as a key broker linking RNA and protein inflammation network to induced COVID-19 severity. This is some of the story of network biology, but COVID-19 network biology shows more results. And by computing, we have published really many, and we have discovered really many important discoveries published in a series of this. This list excludes the International Cancer Genome Consortium and the COVID-19 result. This is only about cancer. Well, this is my concluding message. Pacific Symposium on Biocomputing is a great meeting. I would like to thank the founder of this meeting, and also I would like to thank many collaborators in statistical modeling, oncology, cancer biology, and the Japan COVID-19 task force, and supercomputers. Thank you very much.